morning. Hope you're enjoying the breakfast. We have two great talks today. First will be from Dr. Chandra, and Dr. Raj will be joining us a few minutes late, so he's the second speaker. Thank you. Welcome.
Thank you. It's really great to talk in front of you today and to share about my lab and our research. So it's been a little over a year now since I joined FIU and started my lab after my postdoc. And most of this year I was surprised, but we spent on establishing the lab equipment and processes and validating our previous models. But now I'm ready to talk and share about my past research and some of the things that we're doing now. So my talk is going to be about the modeling the static and progressive neurological outcomes of repetitive diffuse TBI or traumatic brain injury. I'll explain why. So this is one of my favorite things to do in the talk is to scare people, right? So why is this so important? Because traumatic brain injury globally, nearly 70 million people sustain a TBI around the world. So some of the reports did the math, which I don't like doing myself. So they calculated that nearly 130 or more people sustain a TBI every minute. That comes down to 2.2 people every second. And in fact, a British report from Traumatic Brain Injury Association concluded that every second someone, or every 90 seconds, someone is getting transported to the ICU or intensive care unit with traumatic brain injury. So that's why it's so important. And I didn't know that my slides turn automatically, but yeah. So this is the neurobehavioral sequelae of traumatic brain injury, or the list of possible physical, cognitive, and behavioral symptoms that can happen after traumatic brain injury. And the peculiar thing in this sequelae is that there can be an endless combination variations of these symptoms in patients with various types of TBI, with various severity of TBI, that will depend on many factors. And the problem in the field is, clinically and in research, is that there's virtually no possible way to predict who, when, and which combinations of these symptoms will develop at some point in their life. So in particular, in my lab, we're studying several possible neurological outcomes, such as epilepsy, post-traumatic epilepsy, and sleep. And I'll explain why. And to do that, I selected one particular population, which is military. So it's not a surprise, right, that in the military, sustaining traumatic brain injury is very common. But if you look at the chart, you'll also notice one interesting thing, that the majority of these TBI are mild. So why is this so important? Well, because think of it this way. If the TBI is mild, the symptoms most likely would be also mild. So what happens next? After TBI, patients often return to work or their duties much faster than those who have a more serious or severe TBI. So what does that mean? That means that they do not recover completely. And they may actually exacerbate the risk. Because of what? Because in military in particular, sleep disruption or sleep deprivation is very common for various reasons. Not only because of the combat situations or training, but for more routine things. Just the hours in which they operate are typically non-standard, involving oftentimes night hours. In fact, the average time of loss of sleep in the military is about 4 hours compared to civilians. And the reports show that there is approximately 1.6 times increased risk of sustaining a TBI if you are in a sleep-deprived condition. And that makes sense, right? Because in the military, they oftentimes operate complex machinery. So that's making the dangerous environment if you are sleep-deprived. So what are the functional implications of sleep disruptions after a TBI? So as I said, in my lab, we're studying post-traumatic epilepsy. And the phenomenon of post-traumatic epilepsy is this. that there are several phases. There's hyperacute, which is minutes to hours, acute, less than a week, and chronic, over a week, that can take months to even years to develop the first spontaneous seizure. So what can you notice at first? That development of post-traumatic epilepsy takes a lot of time, right? It may take years until it will be established. However, sleep problems, such as sleep disruptions, insomnia, happen within the first months after TBI. So that means that before the development of epilepsy, there are other neurological processes that can influence who and when and whether or not will develop epilepsy. When we're talking about sleep in features, we typically classify two important phases: non-rapid eye movement and rapid eye movement sleep. And epilepsy happens most of the time during the non-rapid eye movement sleep, because during this phase, what is happening to the brain? It's completely synchronized. So epilepsy is a problem of hypersynchronization. So the more that happens, the more risk of epilepsy. And in contrast, REM sleep is a desynchronized state. So it is considered to be anti-epileptogenic state. But the problem with TBI and sleep disruptions is that, again, here's the variations of possible outcomes. So there can be immediate disruptions, there can be variability of sleep disturbances, even in the same individual. Oftentimes, these symptoms are persistent. 25% of population of TBI patients will have insomnia or lack of sleep. 50% will have chronic fatigue and somnolence. And 25% will have parasomnias, which would be abnormalities at any point of sleep stages or their transitions. Now, if we're talking about the problems in the field, what are they? There are problems with clinical and preclinical studies. And most of the time, they are because of very simple things. How do we evaluate sleep? Sleep assessment methods. There's EEG, there's polysomnography, there's actigraphy, there is self-reporting. But in animals, we only can do EEG or behavior, right? We cannot ask a mouse how do they sleep. So that makes it difficult to not only translate studies from animals to humans, but also to compare studies even within clinical studies and also within preclinical studies too. And even if we have the EEG in clinical and preclinical studies to compare, they will often be also limited, because the important features such as power spectrum, information about individual frequencies of EEG is oftentimes omitted or missing. So that makes it difficult to compare what are the quantitative data behind the sleep abnormalities. And when we're talking about the clinical stuff, so oftentimes when people get into the intensive care units, many of them are getting iatrogenic or the other causes of sleep deprivation that are not related to their injury, which are often caused by the hospital itself. Frequent monitoring, often during the night when patients are supposed to sleep. Turning on the light, turning off the light. If any one of you ever was in a clinic or stayed overnight, you would notice that the nurse would come every hour or so and say, "How are you doing?" That's probably not the first thing you would want someone to do every hour when your head was injured, right? So one study called the Siesta in Chicago University is actually attempting to modify this this culture and this monitoring routine in order to make it less invasive, to improve sleep and to improve the recovery. And the other studies have actually calculated that on average, in males sustaining TBI of various severity, they typically spend 8 days in the ICU, in females 6.2 days. So this raises important questions. How do these sleep disruptions affect the risk of epilepsy in individuals? And can early post-traumatic sleep changes predict the development of PTD? So this is what happens. TBI is day 0, sleep disruptions within the first 3 months, as I said, and then they continue, and within 2 years, development of epilepsy, right? So perfect study design. But the problem is that once epilepsy will be established, it becomes a vicious cycle. So one thing promotes the other. The more seizures, the more sleep disruption. The more sleep disruption, the more seizures. So how do we tease apart the role? So this is, in fact, what I proposed to do in the recently submitted DoD grant. We were proposed to investigate the influence of chronic sleep deprivation either before TBI or after TBI to evaluate how it will modify the brain anti-seizure network or mechanisms and will influence which animal will develop seizures, which one will not. So a little about my lab, finally. So there are several directions that we're working with. So as you already noticed, epilepsy and sleep is one of them, and Alzheimer's disease is another, because the more we were looking into sleep and epilepsy, the more correlations and links are with cognitive decline and early onset Alzheimer's there is. And in fact, patients with Alzheimer's disease, oftentimes at chronic stages, demonstrate exactly the same features as in other pathologies. So they also can have seizures, they also have sleep disturbances, and oftentimes one is exacerbating the other. So the model that we're using in our lab is the weight drop, and we're using electrophysiology, transcranial magnetic stimulation, and wet lab. So first was to establish the model, which we did. We demonstrated the seizures. Then we looked at that the majority of animals actually had seizures during the day. This is important because clinically that's also true in patients. In mice sleep during the day, right? So that's an equivalent of humans having seizures during the nighttime. Animals that demonstrate seizures also show that they're staying more awake, they have less of the NREM sleep, and they have more arousals. And when we look at sleep architecture, we notice that TBI decreases the number of bouts of slow wave sleep and increases the variability of the NREM sleep. So this is important because now we can actually find how different are the sleep phenotypes in mice that do and do not develop post-traumatic epilepsy. And I'm going a little over time, no? Okay. So in collaboration with Dr. Boyson from Rutgers University, who established the model of adenosine kinase overexpression in mice. This model has demonstrated that mice with overexpression, genetic overexpression of adenosine kinase, already by default demonstrate spontaneous seizures and sleep alterations. So that made me think that how after TBI this could be one of the mechanisms that links actually the one problem with another problem, and can it be used as a predictive marker? So later we also noticed from his papers that he found additional correlation with epilepsy patients and Alzheimer's patients. So that adenosine kinase overexpression was found in both patient populations in the brain. And then I said, okay, let's do that. So we took mouse tissue, we looked at animals with TBI and without TBI, and surprise, surprise, in the hippocampus, we saw the reactive astrogliosis. The astrocytes turn reactive after brain injury, and they start overexpressing one of the proteins called GFAP. And these reactive astrocytes are those that overproduce or overexpress this protein, adenosine kinase. So what does it do? This enzyme is important in metabolizing adenosine, which is a central neurotransmitter in the brain that is a sleep driver that makes you sleep well. So the more adenosine kinase, the worse the sleep is. The more adenosine kinase, the more risk of seizures. So with this, while we don't have statistically significant data yet, because we just started this pilot study, we already see that within the first 2 weeks after TBI, in the hippocampus, which is the most important structure in the brain that responds to injury very quickly, because after any sort of injury, hippocampus is going to be one of the most vulnerable structures. Why? Because it's mostly composed of excitatory neurons. So even loss of a few inhibitory neurons would be already a dramatic shift in the excitation and inhibition balance in the structure. So our hypothesis was that here's the healthy brain, it sustains a TBI, and what happens, there is acutely increased expression of adenosine kinase, which can be dynamic, but most of the time it's progressive. So over time it becomes worse. And adenosine is a very interesting neurotransmitter. It has a lot of different receptors, as you can see. And how is it related to epilepsy, for example? So one of these receptors, adenosine A2A, for example, is important in the synthesis of glutamate transporter, which is on astrocytes. So the more of this transporter, the better is glutamate transport and homeostasis. The less of this protein, the worse it will be. So the worse it will be, the more hyperexcitable the environment would be and more risk of seizures. So our hypothesis is that the changes of this molecule are related not only to epilepsy, but also to sleep and Alzheimer's disease as an early onset and progressive pathology. So how do we do that? So in the previous work, we used the live mouse two-photon imaging where we studied astrocytes and we were interested in how did they respond after injury. And we noticed that over time, when we did repetitive two-photon imaging, we noticed that astrocytes undergo gradual loss of protein signaling. So this is just a reporter, of course, not a functional protein. But when you do histology, we also notice that they lose the functional proteins such as GLT1, glutamate transporter, glutamate synthetase, potassium buffer channels, connexin 43 that makes the connections between the astrocytes within the brain. So this is a continuous and progressive problem. And in fact, astrocytes would be among the first cells that would respond to brain injury, and neuronal response would be much later, months later. So that's what mimics the clinical situation in context of development of epilepsy, because some features like sleep happen almost right away, but epilepsy, when neurons will become hyperexcitable, happens much later in time. And that makes sense because astrocytes are actually those cells that overexpress this adenosine kinase enzyme. So the worse the pathology in astrocytes, the worse the problem with adenosine kinase and imbalance between excitation and inhibition. So how do we know now that in this model and in astrocytes pathology that it will lead to these imbalances in excitation and inhibition? So one way we looked at that was histology. So we stained the brains of mice with glutamate transporter, and we saw global loss of this protein in the cortex, which would make sense that the environment is more hyperexcitable. But there is also another neurotransmitter, which is GABA, inhibitory neurotransmitter. So I'm interested now to know if the problem is that there is too much excitation, or there's too little inhibition in the brain, or maybe both. How do we do that? So we developed a new adeno-associated virus vector construct using the fluorescent GABA sniffer sensor under human synapsin promoter, with which we first infected the mouse hippocampus neurons that you can see over here that were injected into the stratum oriens, which is the layer where most of the inhibitory interneurons are located. And you can see that it prominently labels these neurons. Now, of course, it is difficult in mouse to study neurons in the hippocampus without removing all the cortex before, right? So that your objective can reach that. So that's not very sounding like a non-invasive procedure, right? So our model, which induces the TBI using the wave drop, it's a closed cap diffuse TBI. Removing the cortex to study another structure would be pretty damaging and would defeat the purpose of doing non-invasive TBI. So logically, next step, we injected the virus into the cortex, where we can image without any lesion. So we can create the cranial window using either thin skull, where you do not remove the bone, but you just thin it to 50 micron thickness so that you can actually see through. Or removing a piece of the bone and exposing the brain cortex and then imaging through. And that's what you can see in here. So with this technique, we're able to differentiate individual cell bodies and their processes, and we can measure the signaling of this inhibitory transfer over time. Now, with ongoing research and new idea, so I'm sharing a little data, but mostly the ideas here. So our new project is actually coming from my past postdoc experience, where we used another model of TBI called BLAST. And that's again mimicking the military conditions, because BLAST is a result of an explosion wave. And it turns out that this causes a dramatically different pathology in the brain, unlike any other TBIs that are contact or focal TBI, for example. So the BLAST differently affects individual brain regions, and they all have different densities. So tissue shearing would be dramatically different also because of the properties of the BLAST wave. So you can appreciate, of course, the size of this device. So that takes probably a quarter of this room and operation of 5 people simultaneously to even induce this injury in a single mouse. And if you're talking about the context of dozens of animals, that's a problem, of course. So we spent several years characterizing this model, and then I thought what we could do to improve it. So as an engineer, you always try to mimic things, right? And make them more suitable and reproducible. So it's a hot topic of tissue on a chip technology, right? And particularly, there are published work already about the neurovascular unit chip model, which combines the neuronal, astrocytic, and vascular elements that you can take from human cells and implant on the chip technology. So our idea is, after we did mimic experiments in the cell cultures, reproducing this phenotype of astrocyte reactivity and astrocytic loss of homeostatic proteins, now we're proposing to design such chip where we can implant human cells and grow them and use our vector constructs to visualize the signaling of important neurotransmitters over time using the 2-photon microscopy. So with this, that's all I wanted to share with you, and I wanted to thank, right now I'm operating on the startup funds and on BME seed grant, and we had a very exciting summer submitting 5 proposals, and we're submitting one in the next 5-6 days to NIH. So we're hoping to maximize our funding opportunities, and with that, expand our laboratory team. So currently, this is our team. We have Naija, who's our master's student, Dil and Pitom, our PhD students, and a great team of undergraduate students from Miami-Dade College. And we are also looking for more students to join from the FIU community. So if you have any questions, I'll be happy to answer.
Questions? Understood everything.
You said that for the imaging, you thin out the skull to roughly 50 microns. How much of a reduction is that? What is the typical thickness of the skull?
Trick question. I don't know off the top of my head what's the typical thickness, because we never actually measured that. We measured mostly what was when you thin it. And there are published work around that, so we'll study bone density, for example, and who first invented this technique. So it's not, we did not invent it, it's been existing for years. Yeah, it's just, it doesn't really matter to which thickness you're actually thinning it. I just used it as a reference to which point. So just keep that, there's still a remaining piece of the bone there. So that's not an open skull. It doesn't become craniotomy, because after you drill through, that becomes a craniotomy. but this is maintaining the integrity of the brain, so you do not expose it to air, and most importantly, what it does not allow for pH changes, because after you open the cranium, astrocytes are very sensitive cells, so any change to the pH will cause them either to die or to become dysfunctional. So this is why using this little or no non-invasive techniques is more important. But it's less about the thickness, because as long as your microscope can image through, you're fine. Yeah, so it can be more, it can be less.
Any other questions? I have, we can talk offline also. But so when you do these, like, you showed at the beginning there's mild cases. That's majority of those cases are mild. And after all the knowledge that you have generated, how do you kind of think about it translates into? Is it some kind of a device or optical instrument? Like, how do we diagnose those mild cases from the knowledge? Is it genetic markers?
So that, how do we diagnose the mild cases?
Yeah.
Of TBI? Well, that's a good point. So what we're doing with electrophysiology, for example, with our epilepsy project, we're looking for electrographic biomarkers.
Okay.
Because we found over the past 4 years that in mice who eventually develop seizures at some point, let's say after 2, 3, 4 months later, if you look at their earlier signatures in the power spectrum, there will be dramatic differences. You will see that there are increases in high frequency oscillations, there are decreases in slow frequency, which would be the frequency responsible for sleep, for slow wave sleep in particular. So the idea is that to promote and advocate for early EEG recordings in humans that would be more comprehensive, for example, an overnight recording. But of course, it's not easy to do. Not all the hospitals have that capacity to do, and not the standards requiring the EEG is usually more severe TBI. So we're trying to change that by saying and showing the data from mice that can be translatable. How serious can mild TBIs be? Because the question is, if even mild diffuse TBI, concussion, can cause seizures, then how different it is from the severe? It only matters maybe how quickly it will be developed, but it doesn't matter as long as epilepsy will be established. So we're showing that it is very important to monitor this population, which is the majority, more than 80% of patient population with TBI, who oftentimes remain unmonitored and undiagnosed. So when things like epilepsy happen, it's difficult to relate it back to TBI years ago. But once scientists now start tracking that over time, they notice more and more correlation that in this population there is an increased risk, and this risk is similar to those with severe.
Thanks. Okay, thank you, Dr. Let's stand as we go. Identify those markers. How do they? I'm trying to see if there's a benefit in mechanical detection. Is that something? Like, even if you say that this particular mind injury causes this mechanics of the property, is it useful? Because nobody is looking at it and exposing the brain and looking at it. Like, how would you diagnose something is going?
It's a very nice.
Besides
relaxing talk on mostly electronics and manufacturing and how it can interface with
So
biology and biomedical
I'm thinking of
devices.
mechanical,
So it's a
mechanically can we say this tissue is gone?
very serious talk, and
Bone
we
tissue,
need
you
to
say
have
this is
a
what happened.
dialogue with each other and make it very interactive, because it's mostly, many of you may find it a little bit remote. So what I do is couple electronics with biomedical devices, particularly electrophysiological interfaces, biophotonic interfaces, and also some kind of sensing for movement or other kind of physical system. So that requires few things. Electronics brings power. You need power for operating everything, right? Without power, you cannot run anything. You also need to communicate, send data back and forth. You need to integrate with the physiological interfaces using sensors, electrodes, diodes, LEDs, some kind of antenna, camera. So basically you need to bring all those interfaces, and so that's what we are all about. It's a very simple logo where we feed data, we supply power, we communicate, and that brings a variety of devices. So it's all about electronic devices and other system components together. So first I will sing a little bit different song about how electronics and manufacturing is so important for all of you. You guys think that biology, some of them are, we deal with tissues, bones, and cells, and all that. But if you think broader in terms of electronics and couple your thinking with manufacturing, then you are a very strong player in the future. So I will talk a little bit different in the first 5-10 minutes. That hope you will be patient enough to see the connectivity with biomedical devices. Otherwise, you will think it's all electronics. Why am I listening to this? So again, we have a very strong set of team, students, undergrads and grads. I don't think any grads are here, so maybe I don't even have to say their name. But undergrad, we have quite a few. They're all here actually. They are also in the packaging class last semester, so they know all these things. They may find it like repetitive. We have Alejandro, Perez, Jose, Ahmed, Asad, Human. So we have quite a few people, and hopefully some more coming in. And systems come with topologies, end user specifications, system architectures, and I am just a conduit. I talk to system guys and bring in many manufacturing electronics and try to realize the hardware with some material process and design innovations. So I am just a conduit with system people. So without the system guys, we don't exist. System guys tell what they want and we make it happen. So we work with a lot of RF system people, biophotonic system people, power electronic system guys, and also new manufacturing innovation experts. So first, very different song. Basically, why is electronics so important? And because in the past, electronics are all discretized devices. You put it on a motherboard, and that's what is happening in the 1980s. But later on, the devices start getting thinner, denser. You need to pack them in a very high density circuit board. Eventually, you want even get rid of the individual device package. You want to use the system package to put all the devices inside, make the devices very thin. And that is what, if you increase the density of the wiring, that is what leads you to future computing, future very high fidelity sensing, where you can integrate extremely high computing power very close to the interface you want. It's very important even for biological interfaces. You want to have some imaging capability with computing. You want to have some high resolution electrophysiological sensors with some computing and communication. So they all come with integrating devices in a very thin package, providing power supply and being able to extract the data, and that is really very important. And we do this across, I only showed it in the context of computing. Actually, the last slide is for computers that are used in data centers, AI, servers and all that. But if you look at it, the same thinking goes even here. So we pioneer a lot of blocks that go into sensors, bring in power supply, bring in embedded devices, some photonic functions, flexible electrode or other kind of interfaces. And many times you cannot put all of them in the same package. So you need to have in the lecture, you guys remember, some of you, a primary package or a distal package, you partition your system, you provide connectivity, some kind of connectors between them. So you need to bring in all these system functions into this very thin package. Same thing applies for computing, for communication, all your 6G networks, 5G networks of the future, power electronics that goes into automotive, and it goes with a lot of building blocks. Realizing a system is not easy, so you can't obviously push 10 building blocks at the same time. So we push one or two and try to realize the system. Like for example, we bring in new power supply integration with a wireless interface, new ways of embedding a LED diode architecture, new ways of connecting these embedded devices in a very thin wearable device. So those are the things we deal with, and you may still wonder why am I singing electronic song to a biology or biomedical audience. And this slide, if you read carefully, it will excite quite a few of you. How many of you are aware of this so-called Chips and Science Act by the government? Basically, in around 2020-21, I think they announced that they're going to invest about $52 billion in promoting electronics manufacturing in US and creating a very vast workforce. They project that US will never be able to supply this kind of manufacturing workforce, and they are very aggressively creating manufacturing research, promoting a lot of startup companies, and massively increasing the investment. So they are doing it through various things. One is called packaging. This is a packaging thing. It's a very big abbreviation, but it's essentially packaging focus where they're putting few $1 billion, microelectronics a few $1 billion, and together they're putting 52 billion. But one thing is here where they're massively investing on workforce creation. And this bill has been approved, and many universities are, partnering with a lot of local startup companies, manufacturing ecosystem, end users. So you have to couple education institutions with industry and the manufacturing ecosystem to be a part of this. So if you can do it and you pitch a very strong case, you can even bid for $100 million overnight. Obviously, you need to have your connectivity, you need to have a killer idea, you need to know who is your manufacturing partner or what is your ecosystem. This is a big deal actually, and it's all about creating future workforce. And a big part of it is creating infrastructure for packaging, which is like what we try to do with our modest labs, and we are obviously very young in FIU. We are only a few years into FIU's advanced packaging lab. But we do our share of this, and we are very aggressive in writing books. I must have written about 25 book chapters, create a lot of magazine special issues, journal special issues. These are the ones I created about 4 years back. We offer a packaging class every semester. Some of you are taking this, some of you are in the last semester. And we organize a lot of packaging seminars with you, with the EMDS, EPS, and the Nanotechnology Council. We host a lot of undergrad students with 3 senior design teams, maybe 1 or 2 in the electrical, maybe 2 or 3 in the biomedical. So they all get exposed to manufacturing techniques, and we write a lot of articles like the way we should be doing. So essentially, it's all about taking discretized devices, eliminating all the packaging interfaces, coming up with very thin embedded devices. This is what Intel and Apple and so on do. If you think why Apple is able to create products, that's exactly what they did 15 years back and what they continue to do. They embed devices, they provide very high density connectivity, they find new ways to provide power supply. So if you think what is packaging all about, it's a lot of words and repeating, but what is packaging all about? It's about enabling the system without being a burden for the system. So the title is very long, but I put all those words. Basically, you want to enable very high data rate without becoming a burden on the power. You want to enable very high reliability without becoming a burden on the size. You want to enable low cost without affecting your performance. So the idea is to make everything zero. I mean, this is very easy to remember, right? We remember zero a lot, which is nice. Zero basically is very easy way to convey what are the blocks we like to deliver power with very zero, almost low impedance. Zero doesn't mean zero. Physics tells you nothing is zero in reality. Zero means a milliohm to micro ohm impedance. Zero means a micro ohm die shift when you embed these dies. Die basically means a chip, a LED, microcontroller, microprocessor. In the packaging world, we call it a die, D-I-E, for whatever reason. So basically embedding these dies with very zero-like die shift, so we can create very high wiring. When you have a lot of components, they interfere. So zero interference electromagnetically. We need to create very fine lines. So we have a few programs where we want to create very precise wiring, zero undercut manufacturing, zero line shift during processing, manufacturing using new monitoring structures in the manufacturing tool. So we have a program and we have a couple of undergrads funded through NSF, and obviously you want zero stress. Nothing is zero in reality. The idea is to minimize to the extent that it is not a bottleneck for you anymore. So one more song on electronics before I go to biomedical devices. So power is a very big thing. We take power very seriously because you need power to do any device, right? You want to extract the sensory information, you want to, excite a photonic device like a LED, anything requires power. So power means basically not simply battery. You may not be able to put a battery. You have to have a wireless link, like a coil or a actuator that can pick up wireless information and convert it to power, or wireless waves and convert them to power. And then that is not good enough actually. Many times you have to have voltage regulators, right? That's very big deal in electronics. Like you guys remember, right? If you suddenly turn on a computer, the lights will flicker. You suddenly turn on your washing machine, your lights will flicker, or you can see that. Why does it happen? Suddenly you turn on a device, why does light flicker? Because you are drawing a lot of current, the voltage line is getting unstable. So there is a voltage fluctuation. So you need a lot of voltage regulators and power supply very close to the device, because I talked about only one device consuming 10 amperes, 20 amps, and still your lights flicker. In a transistor, you have 100 billion, in a processor, you may have 100 billion transistors. Just imagine. Your 100 billion transistors may be drawing a few, microamps of current, but still there are 100 billion. So you may be drawing hundreds of amperes, maybe even thousands of amperes in a picosecond interval, not nanoseconds, not microseconds. So just because you turn on a computer, your lights flicker. What happens if you turn on 100 billion transistors in a picosecond rate? Your voltage lines will all get very berserk, right? They'll go wild. So you need a lot of voltage regulators very close to the processor. And we have, Alejandro and Viru working on multi-layered inductors, and we want to make very high density nanostructured devices to supply the power. So Reshmi is working on it. Eventually we want to print electrodes, and we want to create very high performance voltage regulators. So Pial and Human are working on it, and this is a very big area, and it plays a role even in everything, all the way from computing, electronics, and so on. And we have a lot of industry support. So we do industry support with very high performance computing, and also other applications related to sensors and wearable devices. So they all have different power architectures. So we work with different people. So similarly, they all require vertical packaging. So, and this is about the packaging architecture, but ultimately you need very, very high density power devices. So before I go any further into electronics, we also work on wireless communication systems, and we try to build very complex power modules and wireless communication modules. But let me go back to what we are good at, which is biomedical packages and bioelectronic system integration. So typically, a bioelectronic system is like a very large titanium can with all the circuit board inside and hermetic feed-throughs, wire electrodes, and now they are getting into more smaller electronic systems with more channels of electronic connectivity, like for a retinal implant, for a almost like a deep brain stimulator interface, DBS interface. You want to bring more electrodes, more electronics into this system, so that you can access more sensory interfaces, and that brings a lot of feed throughs, a lot of electrodes. I think that's what you saw even in Dr. Chandra's talk. Very high density electrodes to interface with the physiology, and 3D integration is common to all. So basically, once you do that, then you can integrate a lot of processing, communication with a very high density channel, maybe creating thousands of electrodes and wires through this high density flex integration. But you also need to be able to provide reliability, right? This device may not even go through the very first stage of FDA, because they say, how are you going to provide a very sturdy protection like the way you have it in the pacemaker? So you need to come up with new ways of making sure that it doesn't interact with the body, it is not toxic to the body, and the body, like water and so on, and other reactive accelerated chemicals that cause aging of the electronics, they don't get into the electronics. So basically that's a very important part. The same thing with wearable medical devices. So previously, you think of heart and monitor, pretty invasive in the sense that, it requires a very big electronic module on the top, and so also the other devices that are being very widely investigated by everyone in almost all universities will have a electronic interface that looks almost like a flex PCB or a flex circuit board. We want to make it into a very thin slice of polymer with all the electronics inside the slice. So we call it slice, single layer integration with component embedding. So it's a nice acronym that also conveys the image of a slice, like putting all the electronics inside and providing connectivity. But still it has to be able to deliver your signal, like your ECG or seismocardiogram. It has to be able to deliver your even biophotonic signal, so that it is interfaced with a device that is very common, like a smartwatch or a smartphone. So, but these two are very different worlds altogether. The stakes are very different. In an implantable device, it's about reliability, it's about long-term functioning of to take care of more acute disease conditions. This is more about monitoring and using new multimodal sensing techniques with computational processing, so that you improve your lifestyle, you improve your assessment of your day-to-day health. So they are all very different games altogether. One is cost sensitive, the other is performance sensitive, and one is about easy manufacturing, the other is about connecting with the physiology needs and the end user, and getting easy FDA approvals through the whole process. So they're all very different in their approach, even though I'm putting them side by side. But everything comes with multi-physics, so you need to be good at multi-physics modeling and simulations. If you are, then you can be a lot more value. Well, for example, can you combine a magnetic wave with acoustic resonator and create an electrical function at the end? A lot of physics, that almost every physics is in what I just said. Deal with magnetic fields and deal with some acoustic resonance, which is a mechanical phenomenon, and rectify it and convert it to a electrical usable power. And you need to do it with embedding storage, because ultimately this has to be stored in a capacitor or a supercapacitor and integrated with all the other components, put in a very hermetic package, and you need to then enable the manufacturing. If you get your end user excited, the end user will say, yeah, okay, this product can function, but how will I get it manufactured? So we spend a lot of time even defining the manufacturing ecosystem. So we actually spend most of our days trying to bring the supply chain, the manufacturing tool partners, and the whole process to scale up, so that otherwise it's not easy to get your end users accept the reality of this technology. So we build devices like flexible devices with embedded photonics inside, so you can detect pulse and oximetry. We put very thin wireless power devices on the back. This is actually a power transducer because of its acoustic resonance. It's not just a patch. It can receive power using a very new phenomenon, multiferroic phenomenon that can be used to power LEDs and microcontrollers. Once you receive the power, you can integrate with rectifiers and power your device. And eventually, we are not here yet, but this is from state of the art, where you want to bring in very distributed set of wireless interfaces for interrogating from the brain, or even peripheral nervous system biomarkers, and along with that be able to deliver power very locally using very small neurostimulators, integrate them with catheters so that you can, once you make an electronic device that you can roll, you can integrate them more easily with catheters. So there's a lot of opportunities, and we try not to be coupled with a one specific application. That's a very different approach from typical biomedical engineers. Biomedical engineers marry themselves to one application. We try to be more a broad enabling technology group that can enable variety of things. It all starts with topologies. Without simplified topologies, you can't do anything, right? If you say that I'll take a big flex circuit board and make it into a thin slice, nobody will accept that, because they will say at the end of the day you deal with all these controllers, voltage regulators, and antenna interfaces. How do you miniaturize? You have to eliminate a lot of electrical interfaces. You have to simplify your topology. And this is one topology that we like a lot, which is called passive backscattering approach, which is very minimalistic in its electronics. But we still need a very strong innovation today. In fact, we actually needed it yesterday. But what we need is this diode consumes a lot of power. So we are looking for someone who can simply test a diode pair combination such that I can send very low power interrogating from my phone, and I can receive the electrophysiological signal. This is consuming almost 20 dB loss. So if you can actually put a very thin power supply to this and turn on the diode, then you can actually reduce the conversion loss quite a bit. So that's a very interesting project, and we want it's a very simple idea. You just need someone who has a lot of background in in controls and circuits that you did in your earlier courses. Similarly, we need to rectify and deliver the power with certain pulse patterns, and we also need to have drivers and controllers to drive the photonic system. So we are relying on Yun Kyu to do this. He also works with Dr. Wei Chang Lim, and we are trying to develop some of these topologies. Once you have these devices, then you need to embed them, right? You need to embed these devices in a very thin package. And we have a couple of new NSF awards that focus on this. How to make a very high density package, how to provide the connectivity, and as you are manufacturing them, how do you make sure that the processes are in control? Otherwise, if your chip is shifting, then you can't make high density connection. If your die is shifting, that's a problem. If the lines are also not well aligned or too fat or too wide. So we are basically developing a lot of manufacturing innovations using inline process monitoring. That's what Ahmed Assad is having his NSF REU research experience for undergrads in the new NSF program. So essentially what we do is basically we try to integrate some camera directly with the 3D printing tool. That's as simple as that. So we
5 minutes of quick question answer because it's almost.
Yeah, I will try to wrap up in 4 minutes, maybe. Yeah. So I'm looking at here. Yeah, by 9:58, I will try to wrap up. So basically, in a 3D printing tool, can we integrate a camera so that the camera will keep telling you how to control your manufacturing path? And this camera should detect hidden features, opaque features. Camera is not good at that, right? So what do? You integrate a millimeter wave antenna. Antennas in millimeter wave can see fine features without getting limited by optical visibility. So a millimeter wave antenna is the right approach, and that's the reason why we got this new NSF Partnership for Innovation Awards. In this process, we are supposed to start up a company and make even the company commercializable by the end of the program. That is the vision of the program. So we have industry partners already coupled with us to enable this millimeter wave antenna based inline monitoring. That is the term you have to remember. How do you monitor process inline? And Piyush, one of the grad students, is working on it. And once you integrate with the tool, this is how it looks. Then the whole idea is to start processing, and Ahmed is supposed to be doing it, and we are getting started. Basically, you embed the device, you hold the device in place, then you mold it, and you form your wiring so that you make your embedded photonic system, or you make your embedded electrophysiological sensor system, or even any other interfaces you want. So you need to be embed devices in a cavity. So we use a laser to cut cavities. We drop devices on a temporary carrier. You need something to hold the device, and then you do all the fabrication. Then you remove the carrier, and now it becomes a very thin package that we can use to. So one important innovation is how to bring in a magnetic positioning system. So we are trying to also develop some innovation in how to enable these embedding devices. And eventually, once you do that, rest all is downhill. We can make some nice wireless backscattering antennas. We made some for a neural recording system. You can make a new way of wireless power using multiferroic resonators and embed them inside so that you don't get obstructed by their physical size. When you bury them inside the circuit board, the size doesn't bother you because that volume is coming for free. Because everything needs a circuit board, right? But if the circuit board itself is made of a functional power supply or a functional antenna or something, then that circuit board is not adding any panel. That component is coming for free. That volume is free for you. So you can do a lot of things by putting things on the top, on the bottom, and now we can make very, very, very high quality biophotonic systems. Some of them were actually done with senior design. In fact, I may be not telling you, but most of these things are done with senior design teams, mentored by grad students and obviously companies like Jabil, Beta Blue, and others. And so we basically bring wireless power. Now we are doing wireless seismocardiogram just to detect cardiac mechanical pulses and biophotonics, and so that we can detect pulses, oximetry, and even other tendencies like blood clot. So all these are done through mostly grad students mentoring undergrads and senior design through company partnerships. And we're also coming up with new ways to integrate them into a cyber physical system like a gloves, bringing metamaterials into this. So we have quite a few students working on all these, so I like to recognize them. And Jose, I don't have Jose's picture, so if he's sitting there, I don't know, I'm sorry that I don't find your picture in time. But we are fortunate to have a lot of NSF programs now. NSF funds university industry partnerships, and we took advantage of almost all of that opportunities. We have partnership for innovation, future of semiconductors, ICOPS, IUCRC, they're all NSF through industry and academic partnerships. So we are at least getting some traction because maybe we found the right partnerships. We got lucky. Some DOD, a lot of industry funding, some names I can't reveal because that's and last slide, which is very important. We almost were fortunate to get a senior design recognition in almost every year for the last, every semester for the last few semesters. So I think in the summer we got first prize, fall we got second prize, almost every time. But we get more often second prize, which is okay. I don't mind that. Because first prize is very competitive with all you guys. So anyway, we are very happy to contribute to the senior design teams in any way we can. Thank you very much. Yeah, any questions? I'll be here.
Thank you. Did you guys have a quick question? Yep, there's a question.
What is the role that you see FIU playing in the future of this kind of open space for constant innovation? I see you have a lot of partners up there, and I heard that the goal was to make a company. So is that kind of what you foresee the direction of all of this going?
Yeah, basically FIU has very strong system focus. As an engineering team, we are younger than other universities, but we have very strong system focus. We know what we want to build. Unlike, just like any other university or any college university, we know what we want to build, and we have a very clear manufacturing research. So we should try to bring in that innovation, system-driven manufacturing innovation, so that we don't struggle with just building blocks, but we also know how to put together the whole system, because system drives accelerated innovation, system focus. So we need to couple with AI computing, 6G, healthcare sensor networks, and drive new manufacturing innovations with the right partners. And that is what I was trying to convey actually to a industry consortia recently. So I think exactly what you asked is what we're trying to say here. Accelerated innovation, system-driven manufacturing focus.
Let's, in the interest of,
Thank you.
yeah, let's thank the speaker again, and of course.